Okay, thank you so much, guys, for coming. Today we're doing lecture 8. We're doing blood cells and platelets. We call them blood cells and platelets. Why? Because platelets are actually not true cells. They are pieces of cells, of huge cells called megakaryocytes. So today we'll kind of talk about the life cycle of these cells, especially the red blood cells. And so some of the problems in the module have to do with the life cycle of red blood cells. So we'll kind of go through that carefully, starting with the hemocytoblasts and some of the mechanisms that the body uses to control, stoppage of bleeding, like vascular spasm, platelet plaque formation and clotting. There's some problems with some of the factors and sorting it out. So we'll go through that in a little bit more detail. Okay, so we always start as usual whenever we talk about the subject, the functions of that. We are very familiar with the basic function of blood, which is to move oxygen, and that's where the red blood cells come in, and distribute nutrients. All right, so we are kind of familiar with most of these functions, such as these. All right, so we know that already. So we kind of did this ad nauseum. We know what that is. Processed molecules. This actually is, say, for example, vitamin D. You know how vitamin D starts in S7-dehydrocholesterol, and with UV light, where is it produced? A precursor for vitamin D in the skin. And then where does it go next? It goes to the liver. So that's the function of the blood, which is to transport processed molecules like that one. Okay, and then in the liver, not only it doesn't just go to the liver, another precursor of vitamin D is made, kind of like the second to the last. Then it goes to the kidneys. That's where it's finally activated to active vitamin D, and it's not done yet traveling, because it has to go where now? Where does the active vitamin D go to next? Yeah, so it goes to the intestine. You see, where it started in the skin, it goes through all these organs. Okay, so if I was talking about the function of blood cells, blood, as a whole, in its cellular component, you talk about those things. You don't just say movement of processed molecules. Okay, so that's
So you can use vitamin D as an example.
Yeah, you can use vitamin D as an example. All right, and some of the factors that we will learn, in a little bit, also are trouble in blood, and I'll point that out when we get there. But that's one that goes around many organs. It just keeps circulating until it's finally activated, and then it ends up in the intestines. Okay, so that's that, and of course, transport of regulatory molecules. What are some of the regulatory molecules that the blood transports? Yeah, hormones. Okay, vitamin. Prostaglandins, which are hormone-like substances. Okay, and regulation of osmosis and pH. Blood is where you get all these ions. Sodium, chloride. What else you get? Those, some proteins. Those plasma proteins, and we'll talk about one of them, albumin. Okay. It creates the osmotic pool of blood. Okay. And also blood. We just talked about it. Transports carbon dioxide. And that was in the module too. One of the ways that carbon dioxide is transported is dissolved in plasma, which is an aqueous environment. H2O plus CO2 equals H2CO3, which is carbonic acid, and then it dissociates to bicarbonate ions. Okay, so that's the major way, in fact, by which bicarbonate ions and H plus. There, one is basic, H plus is acidic, and that is also used to control pH. So when the pH kind of goes down a little bit, you know that you've got lots of carbon dioxide in blood. And that's how, you know, blood is involved with that, with all those things, based on some of the other functions it does. Maintenance of body temperature. All right. When blood travels to the periphery, okay, you can lose a little bit of heat. When blood travels deeper in there, you conserve more heat. Because what blood does is that when it goes to the muscles, where it perfuses, the muscles are the ones that create all this heat. And it is blood that actually distributes all this heat throughout the whole body, right? Okay, so maintenance of temperature, because there's no point in the muscles creating all this heat, and there's nothing to transport it to all the extremities like your big toe, your little finger, and so forth. Okay, it is blood that does that, that distributes the heat around, but the heat itself is produced by the muscles, okay, not the blood. The blood just does the transporting. Okay, oh my God. All right, so, you know, of course, we know about the protection of microorganisms, especially the white blood cells. When we talk about that, the leukocytes, okay, we'll have a separate lecture where we just talk about immunity, and we'll really go into detail about some of the processes that the white blood cells are involved in terms of, you know, protecting the body against microorganisms and toxins. So you can give examples of leukocytes here, okay? And then when we talk about the immune system itself, then you can start talking about the processes. But for now, I just want you to be able to say, okay, protect against microorganisms, and therefore leukocytes are involved, like the neutrophils, basophils, and eosinophils, which are the granulocytes, are kind of like, you know, the phagocytes, okay, and they're active in allergic reactions. But then the other ones, the lymphocytes, those are the ones that produce antibodies. Okay, so you can talk about that, and then stop there. Okay. And then when we do the immune system, we can go into detail about it. Of course, clotting. Okay, all the factors in clotting. In fact, we will talk about two ways by which blood clots. The intrinsic way, where something within blood causes the clotting, which is called Hedgeman factor 12. And then something outside of blood, like what is thromboplastin is released by damaged tissue, and that's the extrinsic way of clotting. Okay, so therefore, what is the main purpose of clotting in the body? Why is it?
Blood loss.
Yeah, to prevent fluid loss. You know what I'm saying? Because you could just die just by losing blood. Okay. If you have a traumatic injury and you're losing too much blood, then you go into that shock, and then you then have those positive feedback mechanisms, you know, just losing blood, and then you're pumping less blood, and then the heart is getting less blood, and then it kind of feeds off on itself until you stop the bleeding. All right. So the functions and composition of blood. We know that blood is connective tissue, even though it's not intuitive, that because it's a fluid, but blood is actually made in the bone, which is connective tissue. So blood is a fluid matrix. It's a fluid matrix, you know, where you have the plasma in it suspended the cells. I mean, red blood cells at the end of the day, we'll talk about it in a little bit, are not really cells, because they don't even have a nucleus, they've got no organelles. All they have in it is a bunch of hemoglobin, which is what is used for what? Hemoglobin is used for? Yes.
Gas.
Yeah, you see what I'm saying? Gas transport, yeah, because it does also transport carbon dioxide. Okay. And we'll talk about that a little bit later. But we'll cover more in the respiratory system, but it was in your module too, in terms of the ways that carbon dioxide. So, you know, it's the only fluid tissue blood is. Okay, blood is the only fluid tissue in there, and it's connective tissue for that matter. It's kind of strange, right? It's composed of liquid plasma plus the formed elements inside. Okay, the blood cells. All right. And the platelets, which we said are not really cells because they are pieces of big, huge cells called megakaryocytes. All right. So erythro, anything to do with erythro is red. Eosinophils, those are red. You know, they stain red in acid stain. You know, so those are one of the things that we problems with. We'll kind of go through that in terms of determining the differences between the white blood cells. All right. Because they're different sizes, they got different shaped nucleus, and they got different functions. So we're going to do that. All right. And white blood cells are called leukocytes. Leukocytes, yes. White blood cells are called leukocytes. And we just did we do the lymphatic system? what did we talk about? We talked about B and T cells, or B lymphocytes and T lymphocytes. Okay. Those are leukocytes made in the lymphatic system, or that mature in the lymphatic system. That's all. They're still white blood cells. Yeah. Yeah, and those happen to be the R granulocytes. They don't have granules inside the cytoplasm. And we'll talk about that because that's because sometimes people will be like, oh, what's the difference between a leukocyte and a lymphocyte? A lymphocyte is a type of a leukocyte. All right, and then of course platelets, which are not cells. Platelets are, just pieces. Yeah, they're just portions of cells. And then of course hematocrit is the amount of red blood cells, from whole blood, the percentage of red blood cells in whole blood. Men have higher hematocrit because than women, because and they have more red blood cells than women. Why?
You don't have a menstrual cycle.
But I knew somebody would say that, but that's not it. The menstrual cycle, they lose a little bit of blood. It's not like a lot. It's for about a week. But the big thing is that men have more muscle. Muscle needs oxygen. So you need more blood. We need more red blood cells. Okay? So that's why. Okay, I knew somebody would hit the menstrual cycle, but that's a good.
It had to be the only guy in the room. Yeah. Well, the only guy in class.
No, he doesn't know how much blood he loses, so we can forgive him, okay?
Some guys know about that stuff more than women do.
I do though, but because I do anatomy and physiology. So total blood volume is about 5 liters. Men again have a little bit more, up to 6 liters of blood, simply because they have more muscle mass than women. So they have more blood because of the red blood cells. Okay. Anyway, so we have the bulk of blood is the fluid, right? The water, the plasma. In it, we have the suspended cells, the white blood cells. We get 5 different types of white blood cells, and then we get, of course, all those ions and gases. And then we've got the 3 plasma proteins, the albumin, with globulins, which are involved in the immune system, and the clotting one, the fibrinogen. All right. Which we'll cover at the end. All right, so just know those things. So blood is a sticky, metallic tasting, tissue, fluid tissue. It's opaque. Okay, it's dark red when it's got no oxygen in it. And it's a really scarlet red, bright red when it's got oxygen in it. Okay. All right. So color varies from scarlet to dark red. Okay, dark red, no blood vessels. I think with the Kenneth. So the pH is about what, 7.35. It's neutral, really neutral, bordering on basic, because neutral is 7. Anything above 7 is kind of basic. So it's slightly, ever so slightly basic. Anything below 7 is acidic. So it's slightly basic. Right? From our pH in chemistry and so forth. Okay, so 7.35 to 7.45. The temperature is about 38 degrees. Okay, you might say the blood is feverish, right? But blood is the source of the heat. So when you have heat exchange, you're going to lose some heat. So that's why the body temperature is kind of around 37 C. See what I'm saying? So that's why blood is a little warmer, because it's the source of the heat, and when it transfers the heat transfer is, nothing is ever 100% like we've talked about. There's no 100% efficient machine. So the transfer of heat, that's where you get a loss of heat, and then so the body temperature is maintained at about 37. So don't kind of confuse that with this temperature for blood, because blood picks up all the heat from the muscle, and in fact, blood will kind of regulate a little bit, like we talked about in the beginning, the temperature, because if blood vessels are more superficial, you can lose more of the heat. So depending on how much heat the muscles are producing, you know what I'm saying? So 8% of the body weight is the blood, right? Accounts for about 8% approximately, all right? Since men are on average heavier than women, 8% of a heavier weight is going to be more. right? Because men have more muscles, so they need more blood. So average blood volume 5 to 6 liters, which is about 1.5 gallons for males, and about 4 to 5 liters for females, right? And it's because of less muscle mass, okay, in females. So that's my sign for female, I think. All right?
Looks like a stick figure.
Yeah, that's the universal sign for female. Okay? The plus sign, the male is.
I know, but it looked like a stick figure.
Yeah, okay. The male one is more kind of like male, you know what I'm saying? But anyway. So, we know about blood, plasma, it's pale yellow fluid, it's kind of fawn colored, kind of like hay, right? Containing over 100 solids from all those ions and all those dissolved proteins and whatnot, right? So it's mostly water, right? It's only plasma, it's only fluid matrix, the fluid tissue in the body, so yeah, 91% water. So these proteins now, I need you to know what they are responsible for. Remember when we were talking about osmosis and pH, when we talked about how albumin is involved in the osmotic pressure in the blood. So albumin forms the majority of the plasma proteins. 58%, more than half, is albumin, and is involved in maintaining osmotic pressure. Okay. Sometimes I may say, okay, can you tell me the functions of these plasma proteins? Those 3 plasma proteins, you get 2 points and 1 bonus. Okay. So globulins, about 38%. What are globulins involved in? When you think globulin, you think what? Immunoglobulin, yeah. Immunoglobulin for immunity. All right. So these will be the antibodies, right? So globulin, think immunity. These are the antibodies, and the activate complement is another group of proteins that in blood that will make holes, kind of sort of arrange themselves on the membrane of bacteria and make holes in it so that it lyses and so forth. So complement also helps to fight foreign microorganisms. Okay, the transport, they bind to molecules such as hormones. All right. Why do you bind these globulin proteins to hormones? Why? Why can't you just? Yeah, but why?
Water soluble.
Yeah. Non-water soluble hormones like steroids are fats, right? They can go into blood in an aqueous environment. You can't do that. There are water soluble hormones which are proteins, and we did it in endocrine because you guys are AMP too, right? Yeah, we did that. So non-water soluble hormones like steroids and so forth, those ones, they take to a globulin, which is like a globulin protein, to make it soluble so that it can be transported in blood. All right, and then of course clotting factors. Okay, remember we talked about when we talked about rates of reactions back in ANP1, and we're talking about how size of the particle and usually globulins, especially enzymes, are globular little proteins because they are more active. Okay, so the clotting proteins are globular. Okay, like the fibrinogen in them, so that they can follow the blood and so forth. So, and then come speak of the devil. There you go. Fibrinogen, about 4% of the plasma proteins. Okay, it's the least amount is the fibrinogen, and this is the one that's involved in clotting formation. And we'll talk about the cascade of the reactions that result in formation of fibrin, which is what forms the clot. Let me end. So converted to fibrin, which helps to form. And then other substances like ions and so forth, nutrients, waste products, respiratory gases. Those are the other 2%, the other stuff, right? Okay, so know about the composition of blood as a whole, right? And especially these proteins. Okay, composition of blood again, mostly water proteins. We know about those proteins, albumins, globulins, and fibrinogen. And then ions, of course, of which sodium chloride is the most abundant ions. Sodium chloride, right? Na and Cl minus, Na plus and Cl minus. Remember, I said salt is the major, Sodium chloride is the major salt in blood, and then of course the nutrients, and then of course the amino acids and glucose and so forth, and then waste products, like urea, creatinine, all right, and then regulatory substances, hormones, prostaglandins, and so forth, and enzymes that catalyze rates of reactions and whatnot. All right, so that's all in plasma right there. Formed elements. So we talk about erythrocytes, we talk about the life cycle of erythrocytes. About 4 months, you have new blood cells, red blood cells, that is. Okay, so every third, about 120 days. Because why they are squeezing through capillaries and so forth, so they get worn out. And eventually they are broken down by macrophages in the liver, in the spleen, and so forth, that we talked about when we did the lymphatic system, right? So 95% of the formed elements. Okay, so that makes transport of gases the most important for this blood, right? 95% of formed elements are red blood cells, and red blood cells have a life cycle. All right, and leukocytes, white blood cells, the remaining about 5%, the remaining 5% is leukocytes. And these go down and up depending on your health, okay, in certain conditions or allergic, when you have certain allergies, they are elevated, the white blood cells. Okay. In fact, when you go to the doctor and they do a complete blood count, they can kind of diagnose a few of the things that may be wrong with you, because certain of these white blood cells get elevated in certain conditions. All right, like if you are infected with parasites, you get eosinophils going up. If you are infected, say with a bacteria, the neutrophils go up. Okay, so if you do a complete blood count, you can tell a lot, but about what's wrong with the person. Okay, so only white blood cells are complete cells. Why? Because they have a nucleus, they can hold the other organelles like a regular cell does. So even though we talk about red blood cells, by the time they are mature red blood cells, they got no nucleus, they got no organelles, they're really pretty much a shell containing. Yeah, it's like a lysosome containing hemoglobin. Really, hemoglobin lysosome. Because the hemoglobin is what's involved in oxygen transport and carbon dioxide. Okay, so the 5 types, we said there's 2, and we'll talk about that. Agranulocytes will be the lymphocytes and the
Monocytes.
monocytes. Yeah, that's what it is. Monocytes become what?
Macrophages.
Huge macrophages. They are, phagocytic cells. Okay, and lymphocytes produce the antibodies. Okay, and then granulocytes, and there's 3 of them: the neutrophils, the basophils, and the eosinophils. They got granules in the cytoplasm, right? And we'll talk about each one of those and talk about the characteristics in terms of shape of the nucleus, really, because that differs a lot. And then platelets are just fragments. These are fragments of cells called megakaryocytes, and I'll show you mega. Okay, mega is huge. Karyocyte is cells. And then most formed elements survive in the bloodstream. Okay, for only a few days, especially the white blood cells. Okay, and then they're done. So that's why they form 5%, but they're elevated in certain conditions. Red blood cells are the ones that last for like about 120 days, which is approximately 4 months. Okay, so you can see a blood smear, and you can see a whole bunch of red blood cells, of course, and then white blood cells. These are what? No. What do you think?
As red as a use on.
Well, not really. It's actually purple, this one. But look at the multi-shaped nucleus. These are polymorphonuclear leukocytes, which are neutrophils. Yeah, neutrophils. And then what are these little bitty pieces?
Platelets.
Platelets, yes. Okay. So neutrophils are the most kind of numerous of the 3. Okay. You can see the erythrocytes and then the neutrophils, and then platelets. So you don't see any of the other ones. Okay, so these are them. red blood cells, okay, they've got this biconvex shape, and we'll talk about the significance of that, which it allows it. It's really the epitome of a relationship between structure and the function. Because blood cells, red blood cells are squeezing through all these capillaries and so forth. So it gives it that ability, and it gives it a higher surface area compared to volume ratio is much higher for red blood cells, which is good for gas transport, right? So granulocytes, because they got granules in there. This is a polymorphonuclear lymphocyte. PMLs, like if you do a complete blood count, they write PML on it. If you ever go to the doctor and they do a CBC, complete blood count, you see PMLs most of the time. They don't write neutrophils on there.
You just said segmented.
Or, yeah, sex is another sort of nickname that they use because it's got a segmental nucleus. It's got sex, or bands sometimes they call them. Okay, so that's some of the lingo that the medical, fraternity uses.
And
yeah.
mature.
Yeah, you see that less mature, exactly. So PMLs, polymorphonuclear, that's a neutrophil, and we'll talk about it. Okay, it's got a multi-shape nucleus, phagocytes, bacteria, basophils, they turn blue in basic state, and they release histamine as well. Okay, for allergic reactions. So they launch that inflammatory reactions. And eosinophils, they got that bilobed nucleus, looks like a dumbbell. Okay, and they turn red. Okay, in acid stain. These ones stain purple in both basic and acidic stain. So that's why they're called neutrophils. See what I'm saying? Neutrophils. So lymphocytes, these ones, they have a big nucleus, big round nucleus, a very thin rim of cytoplasm. All right, and we'll talk about that. But you can kind of see that. And then this one, the monocytes, these are the largest white blood cells. They got a kidney-shaped nucleus. It looks like a kidney. Okay, so these are some of the adene parts. If you're like the technician or the CLS or whatever, you've got to know this guy. It's different, be able to identify them. Okay, so. Okay, so the cells that give rise to all these are stem cells, right? Stem cells can pretty much be programmed to become anything. So this line will give rise to, all the 5 white blood cells, the red blood cells, and also the platelets, right? So the red blood cells start off as the hemocytoplasm and proerythroblasts, because that was in your test, and kind of people are prone to that. And I'll show you a picture of the whole life cycle for all of them, right? Okay. So you got hemocytoblasts are the first. They're produced in bone marrow, of course, and hematopoiesis for blood cell production. All right. So in the fetus, it will occur in different parts, blood cell formation, and in the fetus, it will occur in more parts. Why? Because you're making them for the first time. You're making an individual here. But when you grow up, the function of producing blood cells shifts. Okay, it's mostly in bone marrow, red bone marrow, and you don't even have red bone marrow all over the place anyway. So in the fetus, it's liver, thymus, spleen, lymph nodes everywhere, red bone marrow. Okay, and it makes sense after birth. Of course, it will shift, especially to the pelvic girdle, especially the ilium is where you have a lot of red bone marrow. The rest of the bone marrow is yellow, which is pretty much fat in terms of storage of fat. And some white blood cells are produced in lymphatic tissue, which are the lymphocytes. Well, actually, they mature there. They still kind of start in bone, right, and then they mature. The B cells, actually, B lymphocytes, will still mature in bone, but then they will migrate to the lymphatic system, so they kind of move. But it's the T cells that actually mature in the lymphatic system. Yeah, in the thymus. Yes. Very good. Okay, so. And then, of course, hemocytoblasts give rise to all the formed elements, and then that's determined by growth factors. Growth factors are kind of like switches that will say, okay, this stem cell can become this. Okay, this stem cell is going to become this tissue. This stem cell is going to become that. So that's what it is. Determine the type of formed element. because you need a switch that's going to say, okay, become this or become that. So this is what I was talking about earlier on. So pro-erythroblast, then you've got early and late erythroblast, and then at this time it's losing organelles. Okay, so by the time it's a reticulocyte, it has lost the nucleus. All right, and a reticulocyte is not a mature red blood cell. It's just one step behind. So what's the difference between the, which is in your schedule, in your module? What is the difference between this and that in a mature red blood cell? Reticulocyte, and what is lost for it to become a mature red blood cell? No, it's already been lost to become a reticulocyte. No. It was in your module. It was in your. Well, but you would then get it right. Now, what did the module tell you?
I remember.
That's a good one. That's a good answer. Ribosomes. Okay.
I don't remember that one.
Yeah. Well, you don't remember that question?
No.
Well, okay. Now you know that they lose ribosomes because here you got the ribosomes. What's happening on ribosomes, by the way? What do ribosomes do in the cell? Remember what we did in MP1. Yeah. And what protein are you making right now? What protein are we making with those ribosomes? Red blood cells, positive hemoglobin. So you make but that's why you lose all these organelles, even the ribosomes that make the hemoglobin at the end, because you've accumulated all the hemoglobin you need. And then at the end, before it's mature, you lose the ribosomes. You're done. That's why after a while, you then have to recycle that red blood cell, because working and working, it's got no organelles to make any repairs on any of these things. You know what I'm saying? Makes sense? Mm-hmm. This is because it's more specialized that way to transport the gases. So it loses all the organelles. Okay, so know that, okay? In the exam, I might ask what's the difference between a reticulocyte and a mature red blood cell. Okay? Yeah. All right, so there's two lines of these, cells that give rise to white blood cells. Myeloid, myeloblasts, and lymphoblasts. Okay. That's why sometimes you say, oh, you got myeloid leukemia. Then this is the cell line that's affected, and it's dividing uncontrollably to such a point that it will make your blood like sludge. You can't pump that around. That's what will kill you. Okay, and liquid tumors are the most difficult to treat. Because solid tumors you could kind of sort of remove. Do you know what I'm saying? You know, you could resect it. And then it might metastasize fine, but still you have something to kind of aim for. But these ones, so that's why they are the hardest cancers to treat. Lymphoid leukemia, then you have lymphoblasts. So it means it's proliferating into a bunch of immature or non-functional lymphocytes, because something is affecting the cell line. So what does the body do when you've got non-functional lymphocytes? It produces more, thinking like, okay, we need more, and then it keeps producing, and then until your blood is almost like solid. You can't pump it.
Leukemia, you have lots of lymphocytes.
Yeah.
But aren't they immature? Like they don't really work.
The reason why your body will be producing them is that there's something wrong with some protein somewhere, and it's producing non-functional cells. So the body's response is to do what? To produce more. And it keeps producing more until you are flooded with those things.
Takes it out of bone marrow too.
Yeah. Something screwy is happening somewhere. Or maybe there's one protein that's not good because maybe one amino acid is missing. It throws everything out of whack. So that's right. So the myeloblasts gives rise to granulocytes, which are basophils, eosinophils, and neutrophils. And then the lymphoblasts, of course, give rise to the lymphocytes and the monocytes. Then megakaryoblast, of course, gives rise to the megakaryocyte, and then, of course, to the platelets. So you know all the bloodlines, but we kind of concentrate on this one here, because this really does all the, because it's about 95% of the formed elements anyway, right? We'll talk about that. And in the white blood cells, we'll really talk more about their functions and, their structure, because they're different in terms of the cells of the nucleus, the size of the cell, literally. Okay. So, like I said, they're biconcave discs. Okay. Biconcave, two concave, another concave. So that's two. And this is, and it doesn't have organelles. It kind of makes sense too, right? Because after you produce all the hemoglobin you need, you're not going to be needing all these organelles in the cell. It's just going to make the cell more what? If you've got a bunch of organelles, it's not a trick question. If there's a bunch of organelles inside the cell, then the cell is going to be more bulky. It's going to be more bulky, and then it's going to be trying to squeeze through those capillary holes. No chance, man. You see? So it's got to lose weight. So, and the only way to do that is to lose all these organelles. So the only thing it needs is the hemoglobin. Okay? So this is really a specialized cell. In that it's no longer even a cell, because it's not a cell as we know it. Okay? So red blood cells are dedicated for respiratory gas transport. Okay? Such as hemoglobin will attach to carbon dioxide on the amino acid, so it forms carbamino hemoglobin, and oxygen will attach to the heme group, okay, to form oxyhemoglobin. Okay, and so functions in gas transport, both carbon dioxide and CO, oxygen. All right. So, like I was saying, the biconcave disc is the epitome of relationship between structure and function, because that will give it a high surface area as opposed to the volume, which is good for gas transport, and it will give it the ability to be flexible and to squeeze through capillaries, the shape of the red blood cells. All right? In fact, there is a disease called sickle cell anemia, where the red blood cells look like a sickle, something like this. And these people are anemic. Why? Because they can't transport oxygen. So they get tired all the time. Like if somebody says you're anemic, it means you're kind of tired, basically. Because of that thing that you don't have oxygen, then your muscles are tired, they don't have, just because of the shape of the red blood cells, you get a condition called sickle cell anemia. And because that shape is specifically related to the function of the red blood cells. To the extent that if it's different, then it's not going to function properly. Okay, it's not going to do the job. So the concave shape allows them to bend around, be flexible. So after about 120 days, it's pretty worn out. So therefore, it's recycled. Most of the materials in the red blood cells are recycled, and we'll talk about that in a little bit. So gives the erythrocytes their flexibility, allow them to change shape when necessary. Okay, so know about the relationship of the structure of the red blood cells and their function. What qualities does that, or what functional qualities does the shape, that biconcave shape, allow the red blood cells? I need you to know that. And then so there it is. You can see it. Look at how it gets squeezed into this thin little strip here. You know what I'm saying? Okay, and then of course biconcave discs. And therefore, to maintain the osmotic pressure of blood is going to be important, right? Otherwise, if the cells are going to absorb water, then you have problems. Or if they're going to shrink some more, then you got problems. Because they will lose or gain water depending on the osmolarity of blood. That's why it's important to maintain it exactly at the correct osmotic pressure. Otherwise, problems. Because you got cells which are bags of fluids suspended in another fluid. So fluid can move, water can move between the two. So you got to have osmotic conditions here in blood. Very important. So all those functions that feed off of each other that we talked about. And you can see the white blood cells, and the red blood cells, and then of course the platelets. Right? You see the biconcave, cross section over there of red blood cells. Okay. Let me play this one, but I may jump one of the other ones so that you see that quite a bit.
Human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind.
See, that's what I was talking
This phenomenon is called hemolysis.
oxygenation, loss of water, right, and hemolysis, hemo and lysis breakdown. Okay, so that's exactly what I was talking about. So it's very important that you have an isoosmotic solution. So that function we talked about with the albumin, very important, because otherwise everything gets screwed up, right? Okay, so good. Hemoglobin, of course, we'll talk about that and how it transports oxygen and carbon dioxide. The protein is the globin, it's a globular protein made of the two alpha chains and two beta chains. So two of those, each of that kind of chain, that unit holds a heme group in it. Okay, and each heme group can attach to an oxygen molecule. So that one hemoglobin carries 4 oxygen molecules, right? Because each heme has an ion. Because ion is what transports the oxygen. Okay, that's how it's. So heme molecules transport oxygen. Of course, iron is required for that. And when hemoglobin is broken down, iron is really recycled. It goes back to red blood cells to make new ones. Okay, so we talked about the color difference. Oxygenated is like scarlet, bright red, and deoxygenated is dark red, almost like maroon, if you will. Okay, so globin molecules transport carbon, the globin part, which is a globular protein. That's what it is. Globular, being globular really gives it a lot of qualities, and being nimble and being able to, all the enzymes, as well all the enzymes, the catalysts are globular as well. Okay, so hemoglobin is the globin. Okay, the amino acid protein part of it is a globin. It's a globular, protein chain. All right, and it carries. So, if one hemoglobin carries 4 oxygen molecules, and one red blood cell contains 250 million. One of those little cells, man, that's just amazing, don't you think so? 250 million hemoglobin groups. Okay, because so we with 4 on each, then it's 1 billion molecules of oxygen in one red blood cell. Can you imagine that? 1 billion oxygen molecules. You can't even count that. I'd like $1 billion. So you see the 4 globular parts, okay, the 2 beta and the 2 alpha, and then you got a heme group on each with an ion, all right, which is where each one of those will carry oxygen, okay, all right. So that's 2, 4, 6, 8 oxygen atoms. So 8 billion oxygen atoms. You know what I'm saying? So anyway, just kind of put it into perspective. So, okay, transport of oxygen and carbon dioxide. So oxygen is transported in these 3 ways. Okay, most of it, of course, is bound to hemoglobin. About 98% of it, which is almost all of it, and a bit is dissolved in plasma. Okay, again, we can add this in the question. Each hemoglobin molecule also binds to 4 oxygen, right? So oxygen is really pretty much either dissolved or pretty much bound to hemoglobin. Okay, even after hemoglobin gives up oxygen, deoxyhemoglobin still has a bunch of oxygen on it. Okay, still attached to it. It doesn't completely give it up. Okay, so oxygen, carbon dioxide is 3 ways. It's got dissolved in plasma, and in the module you had to rank them in order of which one is the most transport. And then, of course, the most transport is bicarbonate ions. It, because of this reaction here, which is a reversible reaction, and then you get, okay, and that's a reversible reaction again into H plus plus HCO3 minus. Okay, so that's a bicarbonate ion. This is the form in which about 70% of the carbon dioxide is transported, and then of course 23% is carbaminohemoglobin. It's bound to the globin part. Okay, Do you see that oxygen is bound to the heme part, and then carbon dioxide is bound to the protein, to the amino acid field, so that it forms carbamino hemoglobin, okay? Because it's attached to the amino acids, 23%. So do you see the significance? Can you tell me the significance why you have the oxygen attaching to the heme and the CO2 attaching to the protein, to the amino acids on the globin? Why don't they just got attached to the, to the heme group? After all, we got 4 heme groups on it. Why is it? What do you think is? Why do you think that is important? To have hemoglobin attached to carbon dioxide and oxygen at 2 different places.
Maintain the pH.
What happens when you've got 2 molecules vying for the same spot? What do you think happens? Okay, if I put 1 donut here and I say, you guys go for it.
Go fight.
Yeah! So do you want oxygen and carbon dioxide competing really for the same spot? No. The only way to prevent it is that this guy goes on his own spot and this guy goes on their own spot. That's why.
2 donuts.
Because guess what? If carbon dioxide wins, then tough luck for you. See what I'm saying? Which can happen because the cells are busy producing a whole bunch of carbon dioxide. They don't have, so you'd have problems, really big ones. So the only way to do it is to have them in different spots. So I might ask you that, right? What is the significance? Well, why is those 2 molecules transported in different spots on the hemoglobin? So that there's no competition, guys. So well, that's very important. So you don't want carbon dioxide to win, because it just might. This is that reaction I was talking about. The major way by which carbon dioxide is transported in blood is bicarbonate, because this is from your chemistry, it's a reversible reaction. CO2 plus H2O, it forms carbonic acid, and this is the opposite of this. So this is empty cells, right? This is happening, this is empty cells. This is in the lungs, okay? That's why it's reversible. In the lungs, you breathe out this. Breathe out. Right? So the reaction is going to go back. And then this comes out. Okay? So that's how it is. Right? So carbonic anhydrase reversibly catalyzes this reaction. So carbonic anhydrase that way, the cells, carbonic anhydrase back that way in the lungs to breathe out the carbon dioxide. Okay? So that's the bicarbonate ion. So 70% of carbon dioxide is transported that way. Okay, so erythropoiesis, formation of red blood cells. Of course, we start with hemocytoblasts, which then become pro-erythroblasts. Okay, and then there's kind of sort of 2 stages of pro-erythroblasts: the early one, the late one. All right, and then they lose. The late erythroblasts will lose the nucleus to become the reticulocyte, which in 1 to 2 days matures to form the red blood cell. Okay, and then it loses ribosomes here. Okay, and it's about in the blood circulating, you've got about 3% or so reticulocytes, and most of them are red blood cells, mature red blood cells. Okay, but in 120 days, the life cycle is completed of the red blood cell, and then it's broken down. Okay, so reticulocytes become erythrocytes or mature red blood cells when they lose ribosomes. Right. Okay, so these are the circulating erythrocytes, are red blood cells. Okay, so if you have a very few red blood cells, then you become anemic. You're not able to transport enough oxygen to your muscles. Hypoxia, which is low oxygen, all right? And then too many red blood cells. Again, we get into that undesirable viscosity. You know how the athletes, you know what they do? They harvest their own red blood cells, and then just over the 100-meter race, they inject the red blood cells into them. And then so that's the blood cell doping. Armstrong used to do all sorts of doping, you know what I'm saying? And then, you know, so that's why they measure, they take a complete blood count. If they find that your red blood cells have spiked, they'll catch you. So that's also not allowed to do that. But, you know, because eventually if they start overdosing on these red blood cells, then you change the viscosity of blood, and then that's no good.
Damage your heart.
Yeah, and so forth. You know. So that's why they don't allow athletes to do blood doping. You know, they call it that. So erythropoietin, of course, is released by the kidneys. All right. This erythropoiesis is hormonally controlled, and one of the hormones is EPO, erythropoietin. So it just depends on the adequate supplies of iron, amino acids, and vitamin B, folate, and so forth. You need all those things to form red blood cells. Okay, so erythropoietin is released by the kidneys, triggered by. Why are the kidneys releasing erythropoietin for that matter? Why kidneys? Why not the heart? Because the heart pumps blood after all. Why the kidneys?
Well, the kidneys filter all your plasma, so they know how much is in there.
Exactly. That's the correctest answer. You see, they filter the blood, so the kidneys know what the concentration of all these ions are, know the concentration of oxygen, all those things, because it monitors all those concentrations. So it's able to. Whereas blood goes through the heart, but the heart doesn't measure what's inside of the blood. So it won't be good. You see? Exactly. So that's why the kidneys release erythropoietin. And some of the things also that have to do with blood and are released by the liver. Because the liver, again, also does the same thing. It filters, it knows what's in blood, and it can measure all those, So hypoxia will cause you to produce more erythropoietin. Why? Why would hypoxia cause you to produce more erythropoietin? Hypoxia?
Because you need more oxygen.
You need more oxygen, so you need more red blood cells. Okay? And then the opposite is true, right? Okay, so I just want you to know the significance of why erythropoietin is released by the kidneys. Because they measure the concentration. Okay, so this is of course enhances erythropoiesis and increases the red blood cell count, the erythropoietin, right? And then once you increase the red blood cell count, you affect the oxygen carrying capacity of blood, right? Because that's what it does. All right. So decreased oxygen, hypoxia, kidneys, because they are filtering blood. They know the concentration of all these substances within blood. Then erythropoietin will cause more hemocytoblasts to be formed and tend to pro-erythroblasts. Okay? Pro-erythroblasts, because then those will become the red blood cell. And then once you have that, more red blood cells, more oxygen carrying capacity. Okay? Okay, so the lifespan, 120 days, about 4 months, right? 30 days in a month. So all red blood cells become fragile, and they get worn out. They are squeezing through all these capillaries and so forth. So so you can't afford to break them down and excrete all that. So because you got all this hemoglobin, all this protein in it, and then you've got the iron. So all those things are recycled except the heme part. Okay, the heme part forms bilirubin, which is a pigment that then goes into the small intestine, and where bacteria forms the brown color feces. Some of it is absorbed from the small intestine, some of these are bilirubin derivatives, and they go into urine, into the kidneys, they're avoided by the kidneys and give the yellow color of urine. Okay? So that's the only thing that's not recycled. So dying red blood cells, they get engulfed by macrophages in the spleen, macrophages in the liver, they're broken down, all right? And then the globin part, which is the protein component, is turned into amino acids, which can be used. They're used again, all right? And then the heme part, hemoglobin, the heme part, is broken down to the heme group and, into the iron and the bilirubin. So that's all the life cycle of red blood cells. Can you see this yellow? Did you see the brown color of faces? So then there's the brown color of faces. At least you can see that. So the globin is recycled, the iron is recycled, goes back to the bone marrow, and forms in the heme part becomes bilirubin, and then bilirubin is converted by intestinal bugs to a derivative that gives the characteristic brown color of feces. That's why you call it brown nosing. It's, you know, kissing around. Okay, so basically you've got the hemoglobin. You break it down to heme and globin. Okay, and then the globin, what do you do with it? Because I'm gonna probably ask you about the recycling of the hemoglobin. And then you break it down to amino acids. You use those. You use them to build more proteins. All right, and then the bilirubin. The bilirubin is made from the heme. Okay, minus the iron. Okay, the bilirubin is heme minus the iron, because we need the iron to recycle in the red blood cells, you know, to form new red blood cells with it. And then the iron, of course, goes back to the bone marrow. Okay, so these are the things I need you to talk about, right? It goes back to the bone marrow to form, you know, new red blood cells. Okay, and then the bilirubin goes to the liver, because that's why when you're jaundiced and so forth, you get liver problems, because the liver is one that metabolizes all these pigments. If these pigments are not being metabolized properly, they end up in your blood and you lose color. You become jaundiced. In fact, it's one of the things we talked about that affects skin color back in the, although it's just part of the pathological condition. So in the liver, and then after that, boom, 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 when the liver secretes bile to digest fat and so forth, it carries with it the bilirubin. That's how the bilirubin is then transported to the small intestine, where the bugs in there will work on it to form the derivatives. Okay, those derivatives give the brown color of feces, right? Intestines, 7, and then some of those bilirubin derivatives are absorbed back into the blood. Then the kidney gets rid of them, and then goes into urine, forms a yellow color. You know what I'm saying? So, and that's it. So I want you to talk to me about how you break down the hemoglobin. You use the globin part to form amino acids, and the heme part for the iron is recycled, and then the heme forms the bilirubin, which is then worked on in the intestine by the bacteria to form the bilirubin derivatives, which gives the brown color of feces, and then reabsorbed in blood gives the yellow color of urine when the kidneys get rid of them. So that's a nice question to talk about, right? Okay, and this is hemoglobin breakdown. I'm not going to go through it. You know why? Because that's exactly what we just did in the backside, right? So some of these, you know, things, cartoons you can watch on your own, right? So when you're studying, instead of just reading it, you listen to it. It's just a different way to, you know what I'm saying? So that's why I kind of include it in there. So that if we have time, we play them, but if we don't, you know, we can play them. So that we can finish. Alright, so white blood cells, the only components in blood that are true cells, because they got all the organelles in it. Red blood cells lose all the organelles. Platelets, they are just pieces of cells. So they are less numerous, of course, than red blood cells, and they're just 1% total of the blood. And you know, can be, you know, leave capillaries via amoeboid movement. All right, and in fact, when the white blood cells exit the capillaries, what happens? It usually happens in an inflammation situation, and the inflammation, you know, factors, they increase the gap between the endothelial cells lining the capillaries, so they can squeeze out. Because these are like real big cells, right? So they can squeeze out through. So that is the amoeboid action. Two functions: protect against, you know, microorganisms, right? And then they also remove dead cells. And we just talked about how macrophages break down what? Red blood cells. Red blood cells in the liver and the spleen. Okay, so named according to the appearance because of their stain in acid or basic stain, and also the shape of the nucleus. Okay, so the broad name categories are granulocytes, which have granules in the cytoplasm, of which there are 3, and then agranulocytes. There's 2 of them, lymphocytes and monocytes. The granules are way too small to be seen under a microscope anyway, so they don't have granules in them. All right. So, granulocytes, neutrophils, eosinophils, and basophils. Okay, contain cytoplasmic granules that stain specifically red or blue in basic, or in both. Like the neutrophils, they stain in both. So they are sort of purplish in color, all right? They are, of course, larger than red blood cells, and they are shorter lived, Why are they larger? They got all the organelles. And then they're bilobed nucleus, they're lobed nucleus, and then they are phagocytes. Alright? The ones that produce antibodies are the lymphocytes. The rest of them are phagocytes. You see that? Monocytes are phagocytes because they become macrophages. Neutrophils and all of them are phagocytes. Okay, so we have two types of granules. Okay, take both acid and basic stain, so they are neutral. So and they stain purple. Okay, so they give you a lilac color, which is a nice color. They contain peroxidases and hydrolytic enzymes and whatnot, and defensins, which are very active in the immune system. We'll talk about defensins. They are antibiotic-like proteins. Okay, they kill bacteria. All right, and neutrophils, they are bacterial body slayers. So if you have a bacterial infection, guess what is raised in your CBC? Yeah, not Columbia based in college, but complete blood count. Okay, so neutrophils will be elevated. So that's why when you have a complete blood count, you can tell what is going on here. So pretty much pus is the combination of those dead red blood cells and gunk and so forth, and debris, cellular debris. So that forms the pus, the site of infections. Because there will be like a bunch of neutrophils there, and then they die off. So there's a neutrophil. Cells, because of the segmental nucleus, also called polymorphonuclear lymphocytes. That's where you have the PML. Okay, polymorphonuclear lymphocytes. Those are neutrophils. Neutral because they stain both in acid and basic stain. Basophils, they stain blue in basic stain. So they have large purplish black basophilic granules that contain histamine. Basophils are also active in allergic reactions. Okay, basophils release histamine. All right, and they also release heparin, which is the anticoagulant that controls the bleeding and so forth, stoppage of bleeding. So basophils release histamines, which are mediators of allergic reactions. Histamines are inflammatory agents. So, okay, and then of course heparin is an anticoagulant. So that's the function of basophils. Okay, they stain blue. Look at that. It's kind of like a sort of bilobed nucleus, really. You see that? Two lobes over there, and then the granules, of course, stain kind of purple, blue, black, so kind of. Stain. Eosinophils, they are red. Anything erythro and whatnot is red. So stain red in acid stain, they are bilobed nucleus, and you can differentiate them from basophils, which also have a bilobed nucleus because of the stain, right? The basophils will be blue, and this one's blue-black, and this ones will be red. Okay. Red to crimson acidophilic large cause granules lessen severity of allergies. So they secrete anti-inflammatories. So they're involved in allergic reactions because when you have an allergy, you have an inflammation situation and you need to control that. Just like when you are clotting your blood and so forth, you need to control it by those things, like prostacyclin, plasmin and so forth, which was in your modules in terms of how do you control all these clots and so forth. Right? We'll talk about that in a minute. So leads to the body's counterattack of parasitic worms as well. Eosinophils are raised. Back in the day, they used to have and they might probably still do it now. They would infect themselves with parasites, these models, so that when they can eat anything they want and the parasites are busy feeding on them. Yeah, there was one who got infected and it was all over the intestines like this. You die, because you want to be thin and so forth and look ugly. Anyway, but you would see the eosinophils in those models will be raised. If you do a CBC, like they should probably do a monthly CBC with the models to see if they can raise the eosinophils, if they're injecting them, infecting themselves with parasites. They do that, people, to look thin.
They also swallow cocktails.
It's amazing what people do to themselves. To look ugly for that matter, it's not even nice. That kind of condition doesn't look good. And these are eosinophils, you can see that, they stain red in acidic stain. Okay. All right, so agranulocytes, lymphocytes, like the B cells, they produce antibodies. So they lack visible cytoplasmic granules because they're so small, you can see them under a scope, similar structurally, but very different functionally. And they have a spherical, nucleus. In effect, the lymphocytes have a very thin ring of cytoplasm. The whole cell is almost all nucleus. All All right. And then the kidneys, the monocytes have a kidney-shaped nucleus and have the largest white blood cells. These monocytes turn into macrophages. All right. Okay, so lymphocytes account for about 25% or more of the white blood cells. Have large dark purple circular nuclei. Okay, like I was saying, they've got a thin rim of nucleus. Okay, they are in lymphoid tissue, of course. Some circulating blood. Remember, those are the B cells, really, the lymphocytes. B cells. Okay, and yeah. So and T cells are the other lymphocytes. T cells are involved in getting viruses. Okay, B cells produce antibodies. B cells turn into plasma cells which form antibodies, right? And T cells are cytotoxic cells that kill viruses, or that kill cells that are infected with viruses. You see what I'm saying? Okay? That's why HIV goes to attack CD4 positive T cells. Okay? Anyway, so there is a thin rim of cytoplasm for the lymphocyte, okay, which are the B cells, in T cells. And then monocytes, these are the largest white blood cells, 4 to 8%, largest white blood cells with a kidney-shaped nucleus, stained with purple, stain, only U-shaped nucleus. So this is like a kidney-shaped nucleus. So this is a distinguishing characteristic of monocytes. Kidney-shaped nucleus in the largest size. All right, and then once in blood, they differentiate into the exit out of blood. They differentiate when they're in the tissues into macrophages, which are very big phagocytes. All right, highly mobile, active phagocytes. Activate the immune system as well. Macrophages also act as antigen-presenting cells. Okay, antigen-presenting cells. So those are the monocytes. See kidney-shaped nucleus. It's a huge cell. Okay. So just know the differences between all these white blood cells and their basic functions. Okay. And so what is this?
Lymphocyte.
Lymphocyte. No, this is a basophil. Sorry. Basophil. Okay. So this is the exercise we're supposed to identify this. And this is the lymphocyte. Okay. And what is this?
You got those two backward.
This is a lymphocyte. No, basophil is this one. Okay. You see in the book. The lymphocyte is like the thin rim of cytoplasm. Huh?
475.
Yeah, and this is a monocyte, right? And this is a PML, polymorphonuclear leukocyte, and this is an eosinophil.
Where's it shown?
The basophil is the first one. The answer is on the back. The answer is in the appendix. You don't see it there because the exercise was to identify them. This is thin rim of cytoplasm, so that's the lymphocyte guys. Okay, lymphocyte basophil. Trust me. Okay, very good. Platelets. They're involved in basophil is thing. Where did you see that?
I've been doing differential since 1991.
But check the answers in the
Check the
back.
answer in the back. Where is it?
I didn't look.
Still never find it.
Anyway,
I guess it's back in the book.
since we're trying to go, yeah, because the thin rim of cytoplasm, that's the lymphocyte, and the blue one is the basophil. Okay. So they stay blue in the platelets.
Back of the chapter.
Yeah, it's like in the sort of like the appendix at the back there, because it's like an exercise to ID them. So function in clotting, the platelets are involved in clotting, and the platelets are the ones that release ATP, adenosine diphosphate, and thromboxanes. Okay, sorry guys, I'm kind of trying to finish up here. Formation of clots is involved in, all right, and the clots of course stop bleeding, further loss of blood, and seal the wounds. Also prevents bacterial infection when you seal the wound, right? So their granules release ATP and thromboxanes, which excite other platelets, which then also secrete ATP and thromboxanes. Okay, and then so those are the megakaryocytes, and then the megakaryocyte changes into or is broken down into
It is a lymphocyte, basophil, monocyte. Figure 16.8.
Okay.
It is A is lymphocyte.
Okay, I guess. Still corrected. All right. So prevention of blood loss is the platelets to stop bleeding. Okay. So there's three ways. One, vascular spasm, and then you know, you got the platelet plaque formation and the clotting. All right. So vascular spasms occur kind of on a daily basis on your blood. It's just like a damage in a blood vessel, and then you know, the smooth muscle contract, and it's a reflex action, right? And then that can actually even stop small blood vessels. All right. So that's a vasoconstriction, and the thromboxans are released by the when there's the damage to the blood vessel that causes that vascular spasm, and endothelium is released by the epithelial cells lining the capillaries, and then of course platelet plaque formation. The platelets become sticky, they close up, they form a plaque that stops the bleeding, and then of course the clotting, which is a cascade of reactions that I said started by the extrinsic and the intrinsic pathway. All right. So platelet plaques, you have them once you know there's damage to the red blood vessels, and then there's connective tissue exposure. Okay, so those red blood cells, I mean the endothelium cells will release that von Willebrand factor, which will attach the platelets to collagen, right? Platelets are attached to collagen because of that von Willebrand factor. All right, and then they also start to release. Once that happens, once the platelets attach to the collagen through von Willebrand factor, which is released by the endothelial cells, they then do what? They secrete ATP and thromboxans. ATP, adenosine diphosphate, and then thromboxans, which then excite other, you know, cells, which platelets, not cells, but platelets, which then release more ATP and thromboxans. Once they become activated like that, then they will also have a fibrinogen. The fibrinogen receptors are activated. Okay, fibrinogen receptors will cause them to attach to other platelets, which then forms the plaque. Okay, and each of these blood control stoppage mechanisms has a control. Like prostacyclin will dissolve those platelet plaques. Okay, so and then it can seal a small blood vessel, and this occurs many times in a day. Your blood vessels are getting damaged, and they're always getting sealed by these platelet plaques, vascular spasms. So this is it. The von Willebrand factor released once collagen is exposed due to cell damage by those endothelial cells. Von Willebrand factor will attach the platelets to the collagen, right? And then AGP and ATP are released, which excites other platelets. They change shape, and they have these fibrinogen receptors, those red ones, which attach to fibrinogen, and then that allows the platelets to stick to each other. Right? Fibrinogen is what is involved in the clotting mechanisms at the end, where it's turned into fibrin. But in this case, the fibrinogen itself is actually attached to the fibrinogen receptors on the activated platelets, which then become sticky and so forth. All right. So blood clotting or coagulation is a formation of a clot. Okay, through a cascade of reactions, and you activate the conversion of fibrinogen to fibrin. Okay, it starts extrinsically by a process where the damaged tissue produces thromboplastin. It starts intrinsically where when blood comes into contact with damaged tissue, it will activate Hedgeman factor 12 or something also called factor 12. Hedgeman factor, right? Which is factor 12. Thromboplastin extrinsic by the tissues, damaged tissues, and so both of these mechanisms will converge when factor X, factor 10, is activated, and then it becomes a common pathway for blood clotting. Okay. Okay. So what happens is when thromboplastin is released extrinsically by damaged tissue, and Hedgeman factor is activated within blood, They all relate to the conversion of activation of factor 10, factor X, and then together with factor 5, phosphatases, and also calcium, phospholipids, and calcium. This form prothrombinase. Prothrombinase is an enzyme that turns prothrombin into thrombin. Okay, so prothrombin is then converted to thrombin by prothrombinase. Okay, and then fibrinogen is then converted to fibrin by thrombin. Okay, and then you have your final clot. The clot forms, the fibrin is formed, and then fibrin together with thrombin in the presence of calcium ions will activate factor 13. Factor 13 is then the one that stabilizes the clot and so forth. Okay, and strengthens the clot. Factor 13. Okay. All right, and then away from the site of injury, you have anticoagulants. Anything that clots, of course, has a corrective mechanism. We've just seen prostacyclin, which is a prostaglandin, okay, that controls the platelet plugs and so forth. You always need a check mechanism because you don't want uncontrollable clotting. So you have anticoagulants, one of which we talked about, which is produced by the basophils, which is heparin. It's an anticoagulant. And also antithrombin is an anticoagulant that prevents the conversion of prothrombin to thrombin. Okay, so antithrombin and then also heparin, those control mechanisms. So we said thromboplastin, extrinsic factor, it gets activated outside of blood, extrinsic mechanism, and then Henchman factor 12 in blood gets activated in the all convergent activation of factor 10. Then it's now common pathway. You get factor 10, factor 5, phospholipids, and calcium together form prothrombinase. Okay, prothrombinase is going to convert what? Prothrombin to thrombin. All right, and then thrombin is going to do what? Convert fibrinogen to fibrin. Okay, and then thrombin, in the presence of calcium ions, converts or activates factor 13. Factor 13 is one that stabilizes the clot and strengthens it and cross connections that occur. Okay, so that's that, and that's the prothrombin, and then that's the clot formation, and then of course factor 13 in the end is. So sometimes I may ask to show how clotting occurs via the extrinsic mechanism. You start with thromboplastin, or how it's activated via the intrinsic mechanism. You start with Henchman factor 12. Right, and then it all converges at activated factor 10, and then it's the same pathway to the end. Okay. All right. So that's the whole process right there. Okay. Conversion of pretty much fibrinogen to fibrin. It's a cascade of reactions, and although we are studying it in steps, know that in the body it happens simultaneously. Okay, we're just doing it in steps like that, just so that we can see what's happening. But this is all happening kind of simultaneously, okay? All these things. All right, so clot retraction at the end. That's why when you get a cut, what do you see when you get some oozing fluid coming from it, which is serum? Serum is just like plasma, except it doesn't have clotting factors in it, and that's it. So what happens is that when the wound retracts, it will pull the edges of the wound together. It's like almost suturing it. So that's why you see sometimes little creases and little wrinkles on the edge of a wound, because of when the wound retracts, and that also prevents bacteria. And that was in your schedule, in your module 2. It was asking about wound retraction and what are the benefits of that. One, it pulls the edges of the wound together, and it also prevents infections because you're closing it up. Okay, so you know the difference between serum and plasma? Plasma is got all the clotting factors in it, and serum doesn't. That's the only difference, is that serum is the same as plasma, but no clotting factors. Okay, so sometimes I ask about that. And then, of course, tissue plasminogen activator converts plasminogen to active plasmin, and active plasmin dissolves clots. Okay, so we have seen heparin, antithrombin, we have seen prostacyclin, and now we have seen tissue plasminogen activator. It's not the one that actually does the clot dissolution, because that was in your module again. Okay, it is the plasmin that has been activated by tissue plasminogen activator, okay? Because plasmin is one that dissolves the clots, okay? So that's control. Anything that has a clotting mechanism has got to be in check. So I want you to know all those things: the prostacyclin, heparin, anticoagulant, and also the tissue plasminogen activator. Okay. All right. So this is how the tissue plasminogen activator activates plasmin to form. Plasminogen to form plasmin, and it is the plasmin that actually dissolves the clot because it breaks down the fibrin. Okay, and that's it. Thank you, guys. I'm sorry we kind of took a little bit. So I'll give you 5 minutes, right? I'm sorry, guys. Thank you. I appreciate it. I'll give you 5 minutes, and we'll meet up at 5 o'clock. Thanks.